You, sir, come right over here. Let that go. You stand right here by me.
All right.
Okay. You, young lady, come right here in the middle of the table. You stand right here and hold this. This works for 6th graders. I forgot to work for, okay, come with me a little bit. Walk towards me. So we're still put in the middle. Okay, let it go. Now I'm going to stand right here. So, see these 4 people?
Yes.
I want you to, as best you can, we're going to make a circle, because these are the 4 points of the circle. So I want you to all sort of gather in. We're just going to sort of throw our hands out. This is kindergartners.
It's more. It feels more.
All right, this is a 17-foot, somewhat circular, 17-foot diameter circle. This is the size of the largest American chestnut.
Oh, man.
So I want you to have that in your mind as we talk how big chestnuts were before chestnut blight. So can you get this in your head now?
Yes.
How big we're talking about trees? Okay. I'm sorry, you missed out. You have to leave.
Is that measured from the ground or 40 feet up?
4.5 feet up.
Who's going to argue about it?
Okay, so it will be.
They are that big. Yeah.
Dr. Double, do you have any handouts? Any handouts?
We have some stuff up here that Tom's going to do when I'm finished.
Okay, but no handouts.
No. People wanted to know if I had things to hand out. No, it's just my voice that I'm handing out.
All right.
So we have to wait for this to come back up. Just want to take a second. Tom Saielli. So let me preface this that I'm with the American Chestnut Foundation. And American Chestnut Foundation encompasses a lot like the Master Naturalists. We encompass 16 states from Maine down into Alabama. And those states, then those 16 states, are divided up into regions. So we have the New England region, we've got the Southern region. We happen to be in West Virginia in the Mid-Atlantic region. And so Tom is the Mid-Atlantic coordinator for West Virginia, Maryland, and Kentucky. Is that correct?
Yeah, and DC.
And DC, okay, well, I left that part out. But, so he's our coordinator for the American Chestnut Foundation. So there's a coordinator for each of those regions, and Tom happens to be our coordinator. And Tom is an invaluable resource when it comes to all things chestnut. So, there are about 15 of you or so here. I have an hour and a half to talk. Then we're going to transition to Tom for the other hour and a half. And so, if you get bored or you want to fall asleep, you have to use the bathroom. It won't offend me at all if you get up and just walk out the door. So I hope to make this entertaining, but the door's there, so you won't bother me if you get up and leave. Where have all the American chestnuts gone, and can we get them back? So here's a picture from the early 1900s of 5 young ladies around a American chestnut stem. And so, let's see if I can do this. This is sort of a name that a lot of people don't like, the redwood of the East. That makes it sound like the chestnut was the king of the eastern forest. It was not, but it was a large tree, as we just witnessed, by 17 feet. This is a poplar. This is a chestnut, and this is a chestnut. So the poplar was sizable as well. However, American chestnut grows 20% faster than yellow poplar. Now, if you walk out the door here at Pipestone State Park, you've got a lot of yellow poplars. We own property. My wife had a family farm in Marion County, which is mostly yellow poplar. So this is a 20% faster growing stem than yellow poplar, 50% faster than oak. Fastest growing tree species in the eastern United States. So So was it the king of the forest? No, but it was in the kingly realm of large trees. On a good site, American chestnut can put on an inch in a year. So if you take that 17-foot diameter stem,
I think it's okay.
it's probably in the neighborhood of 225 to 300 years old for a tree to get that large. Now we're talking about not only 17 feet in diameter, but 120 feet tall. Think about that, 120 feet tall, 17 feet in diameter. So when you do presentations like this for 6th graders, you say, can you cut that tree down with a chainsaw? Not very well, no. So can you imagine those days with those big saws going back and forth with 17 feet? I can't hardly imagine.
How fragile is it?
What's that?
With that fast rate of growth, how fragile is it?
How fragile is it? It's a really good wood to work with in terms of, we're going to get that into a minute, so I'll hold that thought. Okay, in 1908, more than 10 million chestnut crossties were cut for steam and electric railroads. So here is a tree in 1923 in West Virginia. That's not 17 feet, but that's probably 8, 10 feet in diameter. Now I find this to be an amazing statistic. 1859, chestnut in western North Carolina was measured with a DBH. You all know what DBH is, diameter of breast height? The 4.5 feet off the ground? 10.5 feet, which is a nice sized stem, 10.5 feet. At 65 feet off the ground, the tree's still 5 feet in diameter. 65 feet off the ground, the daggone thing's still 5 feet through. I like to tell people, if you were settlers in the 1700s, and this young man and his beautiful wife decided that they were going to set up shop somewhere in the Appalachians, that you would build your house out of chestnut, you would roof your house out of chestnut, you would use split rail fence from American chestnut to go around the house. You fed yourself with chestnuts, you fed your hogs with chestnuts. It was a tree that built America. And the reason being its properties of having a very high content of tannins. And so in this depiction, with a log cabin of chestnut and a roof of chestnut and split rail fence with chestnut, it was an extremely invaluable species in the eastern United States. Why? You built your fence posts, your railroad ties, you could use furniture. So getting back to your comment, is that there's an old adage of chestnut is that it went from cradle to grave, because you've manufactured your cradles for your infants out of the wood, and you made your coffins out of chestnut. It's not a great wood for burning in a fireplace because it sparks a lot. But in terms of its fragility, I would say it's a great wood because it's an easy wood to work with. My wife and I went down to the Canyon Rim gift shop early this morning, and there are a few, and so down in the basement of the gift shop are artisans, people working with leather and wood and what have you. And so there are a few pieces of wood down there that are wormy chestnut in the gift shop here. Tannin, we're going to talk about that in a minute. Yeah?
Why did it spark? Was it?
I don't know. Do you know why it sparks? I don't know why it sparks. I know I've burned into my fireplace, and you got to stand back because the daggone stuff just shoots out sparks. Yeah, I'm not sure why. I don't know the answer to that.
But maybe a sap or something that.
No, these were dry, so I don't know that that would necessarily be an issue. I don't know the answer to that question. I'll be honest. I could make up something up,
No,
but you know, that's all right.
Is wormy chestnut still available?
Yeah.
You still buy more of it.
Mm-hmm.
Get it from old barns.
Yeah, a lot of old barns. Which is, so that's a really good question, because the piece that we saw in the gift shop was a nice little flyer inside the box that was made out of chestnut, and it was reclaimed from an old barn.
Yeah, I always think it's really interesting when we talk about how many things were made out of American chestnut. And I don't know if you mentioned how rot resistant it is.
Yeah, we're gonna get into that,
All
yeah.
those old things, you know, would still be around. Can you imagine how many old barns and buildings were torn down and the wood was just chucked because people didn't realize?
Mhm. But now you gotta pay a premium price for that kind of stuff. So, you know, shingles, telegraph, telephone poles. So what I tell people is why this built America is that not only did you build your cabin and you fed your hogs with this, as we industrialized and moved west, what did we make our telephone poles out of? Chestnut. What were the railroad ties made out of? Chestnut. And the reason is the tannin content. And so, you know, people think of tannin in terms of tanning hides and leather and stuff to make your jackets and your moccasins and your leather pants in the 1700s. But it's this property that makes this possible 50, 60, 70 years after that split rail fence was put in, because the tannin content is so high. So it's rot resistant. The bugs don't like it. So you can still see split rail fence of American chestnut in 2019 that was put in the ground in 1935. So that's the reason, that's one of the reasons this tree was so important, was because of this high tannin content. Look at this mountain of husks, of burrs, and the bags of chestnuts that are going to market. I often tell the story that I got involved in chestnut research in 1977. And in the early 1980s, we made a trip to Grand Haven, Michigan. And a fellow who's now deceased, I'm guessing back in that era, he was well into his mid-80s. And he had some large chestnuts in Michigan on his property. And when he began talking about chestnuts in his lifetime, the tears began streaming down his face. Because it's such an emotional tie for people that actually remember the tree. Because he said that he and his siblings would go out into this orchard of chestnut trees and collect nuts. Well, they collected burrs to get the nuts. And they put them into bags, and those bags went to Chicago. From Grand Rapids, Michigan, went down to Chicago for being sold by vendors on the street corner with the big barrels and the fire and all that kind of stuff. And it was the money that he and his siblings received from selling his bags of chestnuts that put new shoes on his feet every winter. And he said, without those chestnut trees, we wouldn't have had shoes, or at least new shoes. So there's a real emotional tie for people that remember this particular tree. So here we have the telegraph and telephone poles, and the reason is this tannin content. Yes, ma'am?
How do the tannins compare to the hemlock?
Ah, that's a really good question. And so a lot of places like Spruce Knob, what did they cut their trees on? They cut them down for tannins. So I would say they're comparable in tannin content in terms of those. Those are good questions. Keep them coming. So this is a company out of Canton, North Carolina. And it says here when the mill opened in 1908, it was the largest logging operation in the world. And what did they cut? Mostly American chestnut. And these are sluices. 3 miles of sluices to cut the chestnut on the top of the mountain and sluice it down to the river below. That's how important the tree was, that they would go to that much effort to get these logs down off the mountain. Chestnut was used, extract from leaves and bark was used as a tea for coughing ailments, breathing conditions, arthritis. I could probably use some of that currently for arthritis, but sore throat and sedatives, and like we talked about earlier, it was a primary source of tannins for the leather industry. And so the next slide is that same Champion Fiber Company with nothing but entire trains of chestnut wood. Entire trains of chestnut wood. So let's talk a little bit about the tree. Did it grow everywhere? No, it's an eastern species. Grew from southern Maine down into northern Florida, but you're not going to find a lot of trees in northern Florida or in southern Maine. The predominant range was this Appalachian mountain area, which is a little bit the darker green. Can you all depict that?
Oh, yeah.
And so you've got West Virginia and Pennsylvania are two states that were solely in that predominant range. The adage is, and I don't believe it's correct, but that a squirrel could get into a chestnut canopy in Maine and not leave it until they got to Florida. Well, that's not true. But you've got areas, say now you get over here into the Piedmont and North Carolina, you're going to find some trees, but they're not going to be a lot. And so you can find some areas, even in the dark range, that have no chestnuts. Then there are some areas in that predominant range that maybe 70% of the canopy is chestnut. So it varies from nothing to 70%. And if you average it out, people like to say 1 out of every 4 trees in the entire Appalachian area was chestnut. So 25%. Now think about that, 25% of all trees just here at Pipestone would be one species. Isn't it hard to believe? Now, do we have other species of chestnuts? We do. We've got a couple of species that are Asian, Chinese, and Japanese. We've got European chestnuts. So those are, we're going to talk in simplicity ways, but those are the species of chestnut. This time of year, just about now, you get these long male flowers, the cactus. Do you have any that are expanded, kind of?
I sure do.
The ones I brought are still kind of wimpy. This time of year, within, let's say, another week to 10 days, you get out of your car in a parking lot and smell a chestnut before you can see it.
I got to smell these all the way.
You did get to smell them?
Here. Oh yeah, my car smells like it really big time.
All right, can you pass those around and everybody can take a whiff?
Is that a Chinese chestnut or an American chestnut?
You're going to find that out when I give my presentation.
I'm holding my breath.
So I will just point out, these are the male catkins. They are loaded with pollen, and they're pretty fresh, a little past, so they were best a couple days ago. And if you look closely, there's the female flowers in there, several of the female flowers. So I'll just pass that
Which is
around.
what the burr comes from. So,
Yeah.
okay. So here's a close-up of the female flower here. They're on the same stem as the male catkins. And then once these are pollinated by the male flowers, you end up with those spotty burrs that everybody associates with chestnut, and there are 3 nuts per burr. So I talked last week to a group of prisoners at a federal prison on this. And the guy says, what about those trees that only have 1 nut in a burr? Ah, those would be chickpeas. Same species, same genus, different species, but they have 1 nut. And the same kind of small burr, but there's only 1 nut, and it's kind of round. It's not.
Chinkapin's the same genus?
Oh, chinkapin?
Castania, isn't it?
Yes.
I thought it was Quercus.
And you don't see that.
So did the American chestnut have burrs like that, and the Chinese chestnut burrs?
They're all the same. They all have burrs. They all have burrs.
But the Chinese chestnut burrs are not needles.
Most Southern Americans.
Okay.
Southern Americans. Let me get my bucket. And I don't want to steal any with any thunder from Tom, but I'm going to. All right, so do y'all know Randy up here in the front row? Randy, raise your hand. Okay, so I gave a short version of this talk to the master gardeners in Harrison County a couple weeks ago. And the reason I say that is that in Harrison County, I had an email from a fellow who said, man, I've got some American chestnuts for you. And we really need, this is where we need your help. We need to find American chestnut trees. And I'll get to that, and Tom can get to that in his presentation. Everybody thinks they have an American chestnut tree. And this guy happened to live near Lake Floyd, which is a little housing development in Harrison County. A bunch of houses built around this lake. And so I met the guy in his parking lot, and we hiked up in this mountainous area, or not mountainous, hilly area behind the housing development there in Harrison County. And those trees were sizable. They were 3 feet. They were pretty straight. And so I think, given those conditions, the guy would think, by God, I've got an American chestnut. No, they were Chinese. So let's talk about, I cut these at a farm we have outside of Morgantown. This is a Chinese chestnut. And how it's different, my poor American, it's sort of shriveled up. It must have come out of the water a little bit too much. Here's my American, here's my Chinese. Your first thing is, look at the back of the leaves. These almost have a whitish cast compared to these Americans. Let me walk over here too. This whitish cast, see the whitish cast on these? I'm going to pass these around. Of course, you've already passed those around that are Chinese. But see the whitish cast? If you get your hand lens out, these are all leaf hairs underneath these Chinese chestnut leaves. Now let's take a look at the front. See how they look shiny? Have a bit of a shiny appearance to them. These don't have any sheen to them, these Americans. So let's go over here now. Shininess on the leaves and no sheen on the leaves. The other thing is, my poor American is just so limp. Twig color. Most American chestnuts have a reddish twig color. Look at the color on these twigs. They're green. The other thing is, if you hold these twigs up to the light, you'll see those same leaf hairs on the twigs. There's no hairs on an American. I'm going to pass these around. The other thing I want you to look at is a European. Now I want you to pay close attention when I pass these around. Look at the buds on these Europeans. They're really dark and they're fat. And if you hold a European twig up next to an American, this is a lot stouter than an American. And so I know when they're side by side, it's really easy to tell. When you're out in the woods, you only got one leaf to look at. It's kind of like, well, but in generalizations, these are all generalizations.
So the twig in the upper photo is the American and the bottom is the Chinese.
Yes, you got that absolutely correct. So let me take a couple of these. American chestnut is relatively a longer leaf, and a Chinese chestnut has a tendency to be broader in terms of its expanse. An American chestnut generally has a sharper V at the petiole, and a Chinese has more of a U shape at the petiole. So you all should be able to go out in the woods now and be able to collect leaves, even off the ground, and determine if it's an American or if it's a Chinese. So then when I get your email that says, I've got an American chestnut, I don't want to drive down to your property and find out you got this thing in your yard.
That's all we've got.
Okay, well anyway.
Are the leaf scars the same on both of them, or do...
Leaf scars, I don't know the answer to that. Leaf scars, Tom, are they the same?
You know what? I'm not sure I've ever looked at these.
Okay, so sepules, these little things that come up below the got any sepules on your chestnut?
Lenticels? Let's see.
Okay, no, I don't see any sepules. They're like little things below the buds.
Yeah.
I don't see any sepules on this. The other thing is, while we're talking about buds, American chestnuts have a tendency to have a pointed bud. Remember on those, okay, let me ask you this. Americans, are they red or green?
Red.
Red, okay. So on a red stem, those buds are pointed. On those Chinese that are green, they're rounded. So by looking at twigs on your tree, you're looking for twig color, red or green, hairs or no hairs on the back of the leaves, hairs or no hairs on the stems, right at the very tip. That's how you're going to be able to tell. So when you give me that leaf, you're going to say, I've got an American chestnut.
We don't have to consider European when we're identifying.
Generally not. Generally not.
They're not showing the wild European, you don't see.
Well,
Or in fields.
okay, we're going to talk a little bit about importation here in a minute. Let me pass these around, and you can all look at the dentations on the European chestnuts as opposed to, so I made this easy for you. I put a C, I put an E, and I put an A. So I'm going to start over here. So you can just pass these around as you go. And take a look at these. Now this like this way.
Couldn't we just pluck a leaf and put it in a baggie and send it to you?
Sure.
No,
Well, no, because I really want the twig.
well, also don't put it in a bag.
Oh no, because it gets
It'll
moldy.
get moldy. Yeah.
Just-
Wrap it up
just-
in a tree locator form. Stick it in an envelope. Make it flat. If you don't send it flat, it'll curl up, and by the time we get it, it's all curled up and deformed. It's really hard to get them under a microscope.
But I really like twigs too.
Yes.
Twigs and leaves, because that helps me give a more full determination as to just the leaf itself. Go ahead.
What did he say, put it in?
Okay, so if you put it into a baggie
Yeah.
and you send that in the mail, it's going to take 2 to 3 days to get to me. It's not going to be moldy by the time you get to it. So if you just take a piece of paper, just take a piece of paper, stick it in there dry, wrap it up, and ship it. That's the easiest thing to do.
What about
So
like ironing it in wax paper?
I've never ironed it in wax paper. Have you ever ironed a leaf? My wife's ironed leaves. Well, then that's the way to go then, iron wax paper. See, you learn something new all
He
the
said
time.
no. Wait a minute, he said no.
Oh, wait, Tom says no. Because?
Because I want to be able to look at the glandular leaf hairs under a microscope.
Oh, you're allowed to
I want
burn them
I want
with
a
the
fresh
iron.
sample, unadulterated.
So there you go. No. So I was acquiescing there for a minute, but it, I'm sometimes wrong. Okay, the other thing is, here's the other thing. You call me, you say, I've got an American chestnut. And I say, you have chestnuts? Oh yeah, I've got a lot of chestnuts underground. How big are they? Oh, they're huge. Wrong. That's Chinese. That's Chinese. If you can take your hands and cup them and get 3 or 4 nuts in there and your hands full, they're Chinese. So Americans are maybe a third the size or less than a Chinese nut. Well, we're going to talk about nut size here in a minute too.
Do they taste different, the Chinese? and
Yes.
they do?
Yes, absolutely. Absolutely. Let's talk about one other aspect between American and Chinese. Form. Form. Remember we talked about the fastest growing tree species in the eastern U.S.? Straight as an arrow. Not all Chinese look like this. Obviously the ones in Harrison County didn't look like this because they were 3 feet through and probably 50 feet tall. But in general, it's more of an apple-shaped form as opposed to a timber-shaped form. And so what we're after, and when we get into the breeding, I'm going to talk a little bit in the breeding process. We don't want more of this. We want this. I'm not going to populate the eastern U.S. with a bunch of Chinese apple trees. You know? I mean, we want wood. We don't want... So, we talked about European chestnuts. They were tall, but they were grown mostly for nuts. We're going to talk about that in a minute, too. This is a graph of exotic species of insects and mites introduced to the United States. So we don't have to talk necessarily about insects and mites. We're talking about importation of anything. And here, what happened in the 1830s? We began to have seed catalogs. Oh my God, you could order something from China and plant it in Summers County. And so the advent of the seed catalog, what happens to introduce pests and pathogens? Look at the curve. It just goes crazy. Now, here we have, I told you about this a little bit earlier. Here we have Thomas Jefferson in 1812. And he says, my wants in the article of plants, I still add a little to my former wants, so as to put me in possession once and for all of everything to which my views extend and which I do not now possess. So in addition to being a politician, he bred plants. If you go to Monticello and look at his gardens, today, obviously maybe they don't look exactly like today, what they looked like in 1812, he was a great gardener. And so the idea that human beings have is bigger is better. So by golly, if I can order a chestnut tree that has huge nuts, why do I want to fart around with those little bitsy American chestnuts? Bigger's better, right? So I'm going to import these trees that produce these big honking nuts because bigger's better. And so this is chestnut blight is part of this whole scenario of importation. Now, do we have the Animal Plant Health Inspection Service that guards our borders now in 1812?
No.
No. So by God, the ships came in from overseas, unloaded all that stuff, and what came out of all that stuff? Things that are pests and pathogens. Tree of Heaven. I'm assuming everybody can raise their hand if they know what a Tree of Heaven is in this room. Yes. At WVU, we had a graduate student working on her PhD, and she worked on Tree of Heaven. And she said that, logically thinking, you would think that as a tree ages and gets really old, it would produce fewer and fewer seed. Because it's big now, part of it's dying. You know, just like human beings, the older you get, are you healthier than you were when you were 20? No. So she found out that Tree of Heaven produces more seeds the older it gets, not less. And that one Tree of Heaven, in its lifetime, can produce 50 million seeds. You know how many Tree of Heaven you see on the daggone interstate driving up and down? They're everywhere. One tree, 50 million seeds. So that the problem you face in the eastern United States is that when you have a logging operation on your property, and you're going to fell trees from about 25 acres, if you don't keep after this, the whole daggone 25 acres is going to be Tree of Heaven. All right. Does anybody have any ash trees on their property now?
No living ones.
That's exactly right, because of this. So these are imported, not important, well, they are important. They're imported pests. We've got a lot of this on our farm. You can't hardly beat that stuff to death. Multiflora rose, stuff is tough. And if you drive a little bit further south, you go into the Carolinas, everybody's seen kudzu. I mean, it just overtakes everything. So the whole idea of American chestnuts being small and Chinese chestnuts being big is that bigger is better, so we're going to import that so that we can use that here in West Virginia. You end up with all this.
Is the kudzu related to the Virginia creeper?
No. No. There's mile a minute weed, which is somewhat similar to this, but Virginia creeper is not the same thing.
You could add woolly delta to that.
That's right. Sure. Absolutely. Absolutely.
Destroying the
Dutch elm disease. I mean, you can add all kinds of things to the list.
Russian off.
So this is just a blight. Tom's got some sticks. This poor stick is a year old. I cut it last year, so it's kind of dried out. What came over on the boat from Asia? Rootstock of Japanese chestnuts. Because we're going to graft these with our Americans And what happens was you end up with a fungus on those rootstocks that came in to New York City.
Can I ask you, what was the idea of putting it on the Japanese rootstock? What did they hope to gain? A smaller, more compact tree?
That produce good-sized nuts.
For an orchard type
Yeah.
situation?
So New York City, there's a zoological park, actually a botanical park, in the Bronx. Okay? And so the person, like Randy here, who's in charge of all the trees in the park, in a former life, his name was H.W. Merkel. And Mr. Merkel here sees this. Now this is just a 6 to 8 foot tree. But H.W. Merkel, in June and July, sees flagging on trees that are 60, 80 feet tall. So up there in the crown, he sees brown and wilting leaves. He thinks. That's my impression. It's this. It took him a while to come up with that, but it's the chestnut blight fungus. See this little stick here? It's probably dead. This little branchlet. Any kind of wound on a tree. Bird talons, frost cracks, dead branches, anything that created a wound. For a spore of the chestnut blight fungus to land in that little tiny crack, and then much like a seed germinates, you get the root to come down and then the cotyledon to come up. Same kind of thing here. The fungus grows longitudinally and, more importantly, laterally. So that as the fungus then, on a stem this size, once it gets around to the back and the entire thing is girdled, then everything from here on up looks like this. It's dead. So you take this fungus on a branch or on a 17-foot tree, the result is the same. The tree's dead. And so what happened was that Chinese chestnuts and the chestnut blight fungus evolved together over millennia. And so you, as a Chinese chestnut, you could get the blight, but it wouldn't kill you because you evolved with that over a long, long period of time. Now, you bring that fungus into a continent that has never seen that fungus before, and that's the result. It's no different than the Native Americans when the Europeans came in and they brought in smallpox. The Native Americans died off. They did not have that immune system like the Europeans did for that particular disease. Now, did Europeans die from smallpox? Absolutely. But there were some that survived. And so this whole idea of immunization or adaptability to the chestnut blight fungus happened over a long course of time. So now you take a species that has never seen that fungus, like the Native American who's never been exposed to smallpox, and this is the result. You end up with a dead tree, dead branch, and this then courses over, let's see if I have a slide there. Okay, well, maybe it's the next one. So you take those areas we talked about in Appalachia. There were maybe 50 or 60 or 70% of one species, and they're all dead. The forest looked remarkably dead.
How long does it take for when the fungus first gets on the tree for the tree to die?
It depends on the size.
Okay.
Depends on the size. So for us in West Virginia, with a tree that's this size, probably 1 to 2 years.
And on the big 17?
Well, it'd probably take maybe a decade for that. But that's if you're talking about an infection at the base. Most likely your infection is happening 65 or 80 feet off the ground. And so that whole decline of the crown would happen before a canker would girdle a 17-foot diameter tree.
Now, do insects spread the fungus as well?
Absolutely. We're talking a little bit about that in a slide or two. That's a good question. Prior to the blight, it was projected, remember I told you about that fellow in Grand Haven, Michigan, collecting nuts? 12 billion bushels of nuts annually. So one of the other important things about chestnuts, in addition to the tannins and its ability to stand up as telegraph and telephone poles and fence posts, was its ability to produce nuts, chestnuts, every year. Now, what's our replacement? Some oak species. Do oaks produce acorns every year? No, they do not. Every 2, sometimes, in drought reasons, every 3 years. So you take away a species that produced nuts on a regular basis every single year. If you're a turkey or a chipmunk or a bear, you could depend upon this as a food source every year. If you're out there scrounging around for acorns, we listened to on Metro News a report by Chris Lawrence on the radio, and he does an outdoor kind of thing. And in the fall, he always spends a program or two on massed production for the year. So, do the deer have anything to eat this year, and the turkeys and the squirrels? Is it a good year for massed production or a poor year for mass production? You wouldn't have had that discussion on the radio if this tree was still here. You wouldn't have had to worry about, oh, is this a good massed year? Oh, we have a few white oaks, but none of the red oaks are producing acorns, or vice versa. So it wasn't only a loss of nuts, but it was a massive change in the scenery of almost all of Appalachia. So, maybe a boat that looked like this that came into the harbor in the early 1900s, carrying that rootstock that was infected with the fungus. 1904, spread about 25 to 40 miles a year. And I've seen pictures. So Pennsylvania, early on in the 19-teens, was extremely proactive as a group, and they brought together meetings like this. They called them the Pennsylvania Tree Blight Commission. And so all of you people would be from areas around Pennsylvania, New York, West Virginia, Maryland, and you would gather as a group of scientists trying to figure out what the heck we do with this. And I've seen pictures of people dressed, men in a coat and a tie, out in the forest with a hammer and a chisel, trying to chisel out cankers from trees in 1915. Then there was a thought, oh, well, maybe this won't spread across rivers. Then they had the whole idea of like firefighting now, where you go out and you cut in advance of the front. Well, this would be the chestnut blight front. So then when I cut trees down in advance of the front, then if they didn't have anything to affect over here, it would stop. None of that worked. So you've got, through the 1950s then, the tree was essentially gone. So in the course of 50 years, you went from, remember that 17-foot diameter, down to a few sprouts in the forest. So remember, it had no resistance. Why does the fungus kill the tree? I know you're not mycologists, although some of you may have gone on a fungal foray, which is mycology. The fungus produces filaments. And one of our grad students said they're kind of like straws, and it grows in the tree. And as these fungal filaments grow in the tree, they excrete organic acids out the tips of these filaments. And it's that acid, in particular oxalic acid, now remember this, because this is going to play an important role in about half an hour. But it's oxalic acid that's produced here and kills that tissue. So when you inoculate a little chestnut stem with a blight fungus, let it grow for about 2 weeks, and then cut it away. You have healthy tissue, which is uninfected, and you've got infected tissue. And it's these fans, or these filaments of the fungus that are growing in the bark, in the vascular cambium, killing that xylem and phloem. And so what you end up with are all these little orange bumps on a tree. Those are the fruiting bodies of the stem. I don't want to get into too much mycology, but the fungus produces 2 types of spores. You have an asexual spore and a sexual spore. And they're both produced in those little orange bumps. And I got a slide next that's going to produce that. But the sexual spores are produced in these things that are sort of look like a pear-shaped structure. And these pear-shaped structures have really, really long necks. So in the next slide I'm going to show you, envision taking a scalpel. This represents one of those orange bumps. So envision taking a scalpel and cutting it off here and looking down on the top of that orange bump. For each one of these flask-shaped structures, are these long, like, I call them like pea pods. And there are 8 spores produced in each one of those little fingers. And there are thousands of those little fingers in each one of these little pear-shaped structures. And what happens is, these begin to pulse, and they shoot these things out and up and get picked up by the wind. So, in general, we consider the sexual stage to be a long distance propagule. And I got a slide that's going to show you this a little bit better. And the asexual is more of a localized spread. Okay, so here's that picture I had you envision in your mind. Taking a scalpel and cutting one of those orange bumps right off the top. See those little black dots? Those are those necks of those sexual fruiting bodies that are coming up through this orange material. And here they are. So if you take a needle and a stereo microscope and you put this under the scope with that needle and you focus down and you take that needle right in here and pop that whole orange thing up and you clean off the material, these are those pear-shaped structures. And these black necks have been cut off a little bit. But these are the things that grow to the top of these and then shoot these spores out. So the sexual spore is a 2-celled spore, which is much larger than the asexual spore, which is smaller. Now that asexual spore is produced in these long, we call them cirri, but this is real sticky. And they're producing like these little pigtail things that come out. And so as the rain washes over this during a rain event, it washes all these spores down to areas below the tree. The fungus is really, really easy to work with. And the fungus is orange when you get onto a petri dish. So it's pretty easy to work with the fungus. You take a knife, or in our instance we use, let me get to that later on. I don't want to get ahead of myself. This is chestnut in American forests today. So when you go to your hometown, wherever that might be, if you have an area that has chestnut, it's going to look like this. You're going to have a main stem that's maybe 4 or 5 inches. It's dead. You've got sprouts. And this is one of the reasons why the tree is not extinct. The fungus doesn't affect below the ground line, where all the other fungi and bacteria are acting in concert, plus there's a lower oxygen level below the ground. All fungi are aerobic organisms. They need oxygen. So when you get below the surface in the soil, with all the fungi and actinomycetes and bacteria and a little bit of lower oxygen, the fungus doesn't survive down there. So chestnut is a prolific sprouter. So this tree dies and sends up shoots. Well, some of these shoots, from my vantage, over here, are still healthy. What do we have right here? Can you pick that out? You have a canker. So that sprout is not too long for the world. So this whole image of chestnut now is cyclic. Is that if you go into a forest and you clear cut, let's say you've got 50 acres you're going to clear cut, and if you had chestnut on a property, maybe they've been surviving as little 4 or 5 inch seedlings on their forest floor, you go in there and give them light, this is what's going to come up. That seedling right there, it's going to grow once it gets light, it'll be 4 or 5 inches, blight free. Then blight comes in several years after, this gets knocked back down, your sprouts get knocked back down, your locusts and your maples begin to grow up, they overshadow this, and sort of relegated back to that little thing on the forest floor. So it's a real cyclic event of birth and rebirth in terms of chestnut in the eastern United States.
Do we have any groves of old chestnut?
I'll talk about that in a minute. So why was the chestnut-like fungus an effective pathogen? We had a very uniformly susceptible host, we had 2 spore types, and a relatively contiguous range of one species. So in my next, I only have a half an hour. We're going to talk about 3 ways to try to combat chestnut blight. We're going to talk about a virus-infected fungus. We're going to talk about breeding. And we're going to talk about transgenics. So I have to speed up here if I'm going to get through the rest of my story. Resistant Asian chestnuts, Chinese and Japanese. Europeans are susceptible, Americans are susceptible. Let's talk a minute about European chestnut. So put yourself back in the early 1900s, late 1800s, early 1900s. If you are a scientist working in Zurich, and you are an expert on European chestnut, and you are a scientist in the United States, and you're talking about chestnut blight, what you have to do is to do research, write that article up, send it off to a publisher, he edits it, sends it back to you, then it gets published. So now you have an article that's published in a journal. And then it has to get across the Atlantic Ocean to him over here in Zurich so he can read about what the heck is happening in Eastern North America. So that's how information, I mean, we live in an era now where information is like instantaneous. You know, somebody kisses somebody, well, you got an Instagram and woo, it's all over the place. But 120 years ago, you had to wait on that information to get across the pond for him to read that in Zurich and find out, my God, their chestnuts in Eastern North America are dying from a fungus. And so they begin to wonder, what's going to happen to European chestnuts? Are they going to be susceptible like Americans? Or are they going to be resistant like Chinese? They don't know the answer to that because they don't have the chestnut-like fungus. Okay? They found out in 1938 in Genoa, Italy, which was their first flagging of European chestnuts. They're susceptible too.
You're in trouble if you ain't calling it back.
See, he's in trouble up there. He probably has chestnut.
No.
Okay, in the 1950s, Giovanni Batziger,
Keep going to the left.
Swiss plant pathologist, hiking in the Alps. Now, let me give you. I have too many stories to tell for an hour and a half. Let's say you're all Europeans, and this table is one little valley of Europe. That's another valley of Europe. This is another valley, another valley. Every one of you had your own specific variety of European chestnut. And yours was a little different than theirs, and yours was different than theirs, and yours was different than theirs. My wife and I have been to meetings where a room 3 times this size was full of bushel baskets of European chestnuts. Different colors, some had striations, some didn't. Yours were used for confections. Yours were used to take the chestnuts whole and glacé them so you can have a great big huge confection. Yours were used for grinding up for bread products. And so everybody had a little bit of different variety of European chestnuts. And of course yours were better than theirs, and theirs were better than yours, and yours, you know, so the whole thing was sort of a competition of sorts. Everybody liked their cellar chestnuts, they were different things. Anyway, hiking in the Alps. This guy's a professor, Botziger. He doesn't see this. Okay, so let's get back to European chestnut. They were grown mostly in orchard situations. Now, were they squatty like Chinese chestnuts? So they were pretty tall. And my wife and I have seen 78-year-old women out there with wooden rakes in October raking chestnuts off the floor, but these trees were probably 60 feet tall. But they were still orchard-grown trees for the nuts, not the wood. Here's a European chestnut. Does that infection look like this? It does not. This is very superficial, meaning it's not very deep. This goes all the way to the vascular cavity, all the way to xylem phloem, kills that tissue. Not here. So, hypothesis. The tree has mutated. That was the hypothesis. There's been some kind of mutation in European chestnut that made this possible. That hypothesis was wrong. But it was a hypothesis nonetheless. Remember I said it was easy to work with this fungus? You take a knife, you chisel out a little piece of bark, you stick it into a container of 10% Clorox, 9 parts water, 1 part bleach, 10% solution. Stick the little piece of bark from your infected tree into that 10% bleach solution, let it sit there for 15 minutes. Take it out, let it drip off. You don't rinse it. You put it onto a petri dish, and the fungus grows out. It's easy fungus to work with. Remember, I showed you this earlier. It's an orange pigmented fungus. That's not what they found from those trees with that blocky bark. They found this. So the idea was, this isn't chestnut blight. It's some other fungus. That was wrong too. This is virus infected. Now think about that. We have a virus infected fungus. Got that? Virus infected fungus. Now I'm going to talk about this. So if you take the orange fungus, you take these are chestnut stems. Take a little piece out, put an orange piece in here, take a white piece in there, cover it over with tape, let that sit in a Tupperware container for 3 weeks. How far has that fungus grown? The orange one? The white one? My analogy to Randy is that Randy is a runner. And Randy, when he is healthy, can run a 4-minute mile. Can't you, Randy? Absolutely. Still can. You infect him with a virus. Doesn't kill him. But he's got a fever of 103. He's had diarrhea. He's vomiting. Can he run a mile in 4 minutes? No. It may take him a half an hour to get around that track for a mile. It doesn't kill him, but it sure as heck slows him down. That's the same analogy with this virus that's in the fungus. Is it doing a 4-minute mile? No. It's only growing this much. So we call that hypovirulent. It's less virulent than the virulent form. This is white, this is orange. So hypovirulent, less virulent than the orange form of the fungus. So, this was found in France, that hypovirulence issue, and they send that to the United States, to the folks at the Connecticut Agricultural Experiment Station in about 1974 or so. This is an article that came out in Science magazine, and everybody thought, my God, with hypovirulence, chestnut blight has been solved. I started working, and so 40 years later, we're still working on that solution, but this led people down the path to think, we've got it kicked. And look at this. This is an article right here on that Science Journal written by Robert Frost in 1936. Now remember, chestnut blight came in so this is the middle of chestnut blight, okay? We hadn't discovered hypovirulence yet. Will the blight end the chestnut? The farmers rather guess not. it keeps smoldering at the roots and sending up new shoots till another parasite shall come to end the blight. How prophetic was that in 1936? See, if I had a clicker, I could go faster with this.
Did he write any more as to why he believed that?
No, I don't know what that's a good question. I don't know the answer to that. The fellow who found hypervariance is this dude here on the left, Jean Grant. He's a mycologist from France. And he's the one that went and sampled that bark and brought it back and found that white form of the fungus. And he's the fellow that did a lot of other things in terms of us figuring out it. So, this is Europe today. Well, at least this is Italy today. Chestnut blight is no longer an issue in many regions, not all. In many regions of Europe, chestnut blight is no longer an issue because of hypervariance. So all these trees, they're all chestnut, European chestnut.
But they're growing slowly.
No, they're growing vigorously. See, they're using these for poles. Some of these logs up here are 3.5 feet through that they're using for furniture and a lot of pole production because, you know, they grow a lot of grapes and stuff in here. So all these poles now, because they go in the ground because of the tannin content, you can leave these poles in the ground a long time. So we were told that when you cut trees in Europe, you've got to leave 10% behind on the forest floor to regenerate the forest and not to deplete it from nutrients and what have you. So lots of European chestnut production. Okay, so what is hypervariance and can we use it? Okay. You're not interested in this, but these little dots, little circles, a double-stranded RNA virus is inside this little lipid. Now, we're talking about Randy getting a virus over here that made him sick, some kind of stomach flu, polio. Those are almost all protein-based viruses. They have a protein structure. The chestnut blight fungus, or the virus, is different. It's lipid or fat-based. So all these little circles here are little balls of fat. So they're very, very different than these protein-based substances, or viruses. So we found a brand new family of viruses that are encapsulated in a little globule of fat. So what happens? We have down here, now these are filaments of the fungus, remember? Fungal filaments. We have one that has, this is an orange filament, that's a white filament. What happens in nature or on a petri dish is sometimes you get fusion of these two filaments. And the little dots are the virus that then get transmitted from the white to the orange. Give me the next slide. Next slide. Okay, so you take two cubes, orange, white, put them on a petri dish. 4 days later, you don't see any difference? That's fine. So right here, can you see the change from orange to white? This is what happens. Those filaments fuse. That virus was transmitted from the white to the orange, and consequently, all the growth from there on out is white. Same thing here. Right here, we have a change. This is all white. Over here, you don't have any change in morphology of the fungus. White, orange, there is no change like this. This phenomenon turns out to be why the savior back in 1975 didn't happen like we thought it was going to happen. The fungus has different compatibility types. Another analogy for humans. You've got A, I've got B blood type. Can we go to the hospital and transfuse you to me? No, we can't. Same thing with the chestnut blight fungus. Instead of having A and B and O and AB, there are 64 types of the chestnut blight fungus. Now, if we're the same type, do those filaments grow together? Absolutely. If they're different compatibility types, do they grow together? Chances are they do not. So what happens is those filaments grow toward one another, and there's a gap. There's no There's no meeting. It doesn't grow like this, it grows like this. So with 64 types out in the field, here's a close-up of these two plugs. See, we have an incompatible reaction here. We have an incompatible reaction here. This one's compatible. Now, if you each represented one tree in Summers County, and you each had one canker on your person, if there were 50 of us here, there would probably be 30 to 32 different types of the fungus amongst those 50 cankers in West Virginia and most in all of Eastern North America. Okay. I took a lot of these slides out. It's controlled by this ability is controlled by 6 genes. You don't necessarily need to know that, but there are 64 types. To be really compatible, you need to be identical in all 6 genes. Go ahead. So, let's get to your question about, who had the question about stands of chestnut? Okay, this is in Wisconsin. If you go back to the map of American chestnut, this is not in the natural range of chestnut. It's well beyond the natural range. The natural range, as we went west, went to just about Detroit. And what's a natural range? A natural range is where squirrels and chipmunks and stuff disseminate the seed without the intervention of man. Simple as that. So this is beyond that. So as a farmer from Pennsylvania, these are actually true stories. Pennsylvania farmer, sometime in the 1860s, 1880s, decided he's going to take his wife and kids, and they were going to head west. And so they headed west, and what did you do in your wagon? You took your apple seeds, you took your favorite pear seeds, maybe some cherry seeds, and by God, you threw in a few chestnuts in that bag as you stuck them on the wagon, and you went west. Okay. So, this is an area outside of La Crosse, Wisconsin, not too far from the Mississippi River. Go ahead. All these little white dots, you planted 9 trees from those nuts you brought from Pennsylvania. You end up with 5,000 stems 100 years later. Nice cornfield. The forest is full of American chestnut trees. The largest stand of American chestnut in North America. So let's get back to you. You're the owner of all these trees. You're an engineer on the railroad. Your name is Ron Bachenauer. And Ron Bachenauer owns this stand, but on your day job, by God, you're traveling up and down the Midwest with freight. 100 years, your stand is blight-free, just like before blight in the eastern United States. So your trees are yay big. They were planted in the 1860s, 1880s. Blight was first found in 1987. Why? My opinion is, as a master naturalist, you're interested in the largest grove of anything in eastern North America. So, what happens? This gets disseminated amongst naturalists. So this young man here says, by God, I'd really like to see that largest stand of American chestnut. So he goes knocking on your door. You're not home, your wife is. He says, I'd really like to see those chestnuts. Don't stomp on the corn. The house is right here. Take a look at the trees. She wants to see the largest stand of American chestnut. He takes her up on the wagon behind the tractor. Says, don't step on your corn on the way out. Where are these people from? Chances are they're from eastern United States. Have they been in the forest in Pennsylvania, in Maryland, in wherever? Could it have been a bird? Yes. My impression is, with all the people knocking on your door, and you taking them up on the tractor to look in here, because the state champion tree takes 5 of us hand in hand to get around. 5 of us to get around. Chestnut blight discovered in 1987. So what happened was, damn, I'm running out of time. hang on 15 minutes, I got a whole lot of crap to cover. But you, as an engineer, have two people come to your door. This guy here says, I think we ought to take an ecological approach and let nature take its course. This young man over here says, by God, I think you ought to try that hyperveillum inoculum stuff and see if it works. It's like you going to the doctor and one guy says, I can treat your heart with medication. The other guy says, by God, you need a heart transplant. Hell, you're not a doctor. What do you believe? Well, you opted for this ecological approach first. You waited several years and all you saw were dead trees. So then you switched gears to this guy over here. And so what happened was that we began numbering trees as they became infected and numbering cankers on these trees as we went up. Some of these trees have 25, 30, 35 cankers up to 65, 80 feet. Go ahead. I got to rush through this now. So we treated these trees. I grew that white form of the fungus up in the laboratory in lots of petri dishes, mix it up in a whirring blender to the consistency of applesauce. Stick it in these plastic containers, take a hammer and a leather punch, and you ring around the margin of a canker. You squirt this juice in there that has the virus in it, and we did this thing off of ladders. Go ahead. We did this thing with 3 professional tree climbers 65 to 80 feet off the ground. They're sampling cankers, they're treating cankers. So here they are up in the canopy. Go ahead. This is what we're hoping for. If those filaments were compatible, and you get virus here and no virus here, and they grow together, you get transmission of that virus from here to here. If that happens, the tree, that fungus slows down, and the tree can use its own natural defense mechanisms to form callus tissue. You wait another 10 years, that daggone callus is going to close up, you won't even see that anymore. So that's the best case scenario of using hypervirulence. Go ahead. How do we know if it's moving around? We use bone marrow biopsy instruments like you use in the hospital to suck bone marrow out of your femur. Makes 2 millimeter holes, nice strong stainless steel shaft, 2 millimeters. We put those into a 96 well microtiter dish. It's a plastic dish. I use a index card on the top. We take 12 samples out of each canker. All these samples out of the same row of the same canker. So you can label the index card tree 116 canker 1, tree 121 canker 2, tree 115 canker 6. So these are all labeled. We cover them over with tape after they're full, bring them back to the laboratory, stick them in the freezer. Take a screen, take that tape off, put that screen on there, rubber band it real tight, stick it in that 10% Clorox solution, 15 minutes, take them out, put them onto a petri dish, daggone fungus grows out. Is it the only fungus that grows in the tree? No. Here's the chestnut blight fungus, but this is another wood-inhabiting fungus as well. Next slide. So, you can already tell, no virus, virus. No virus, virus, and things that aren't chestnut blight. So we have all kind of stuff coming out of the wood of these cankers that we've treated, with and without virus. So we're doing this from 1990 up until the year 2000. In the year 2000, we had 500 infected trees and 1,600 cankers. Well, we're a group of people from West Virginia, from Wisconsin, from Cornell, from Michigan State. There's maybe 25 of us. Hell's bells, can we look at 500 infected trees with 1,600 cankers? No. So, next slide. We divided that stand-up into areas where the disease center was. All these trees are infected. These trees are somewhat infected, and these trees are hardly infected. Remember again, here's the house, here's the cornfield. So 95% infected, 30%, 10%. This is in 2001. Okay. We divided up into plots, disease center beyond the front. Each plot's got maybe 40 to 60 trees. A little bit of data. So in 2001, when we treated cankers, okay, so the other thing is we treated about 2/3 of the trees in each stand, in each plot. Left a third untreated so we can look for tree-to-tree movement of the virus. So in 01, on our treated cankers, it was about 1/3 of each. Virulent fungus, the orange one. The white fungus is blue. The green is fungi that aren't Cryphonecter. 2016, 15 years later, the pile looks pretty much the same. because we're treating these cankers. These are ones we're actually ringing with a hammer and a punch and sticking a goop in. These are those trees that weren't treated. In 2001, most of that pie shape is orange, or red. That's the orange fungus with no virus. 2016, 15 years later, that stuff is spreading quite well. So we actually have more of the white fungus on the non-treated trees than we do on the treated. So the bottom line of this story is, these two trees were probably treated in 2001 or so. This one responded well to the treatment. This tree died. So it's sort of a mixed bag as to, I could talk another hour and a half on this, but you keep going, because we've got a lot of ground to cover. We took the white fungus, and we talked about those 6 genes, and there's 64 types. Make a long story short, we knocked out all those genes that made this a function, and knocked them out with the hopes that it could be this with any of the 64 types. I know that's a quick description. We call this a super donor fungus.
How many of those? How many of the 64 were super donors?
2. With the hopes that they could interact with all 64. Go ahead. So this is in Western Maryland. Once again, we've got trees in Appalachia look like this. Some of our larger trees look like that. We had 3 sets of plots. One where we used the super donor fungus, one where we used the white fungus with no knockouts, and then water auger. Those are our 3 treatments. Go ahead. Here's the knockout fungus. It looks a whole lot different than this. These genes are knocked out. It still grows, but it grows really slowly compared to the orange form. If you take a virulent form, the orange form of the fungus, and on either side, that white super donor, let those things grow for 2 weeks. There's no control here. This is water auger. And look at the control we have with the super donor fungus. It's the same here like we showed a couple slides ago with the orange and the super donor. All this growth from here on out has all been virus disseminated. So what do we do? Again, we use that punch. Go ahead, we're going through these fast. Put the goop in. Super donor fungus after 2 years. This is actually 3 years. Really good control. See all the callus tissue here? Just walling that off. Another good callus tissue. This is water auger, and there's absolutely no control whatsoever. So the super donor fungus works pretty well. One more. Here's a little bit of data. This is 3 years after treatment. See that callus tissue beginning to close up? So it actually has a significant difference in canker expansion from the super donor to the white fungus to the water auger. Okay, I can talk about this for another half an hour too. I got 10 minutes. I got a lot of stuff to cover. Okay, so you and I are having lunch at a meeting in Eastern Hungary, and we decided we'd both been to a talk by this guy here. And I had lunch with this woman, and she said, my data doesn't really agree with his, and I said, neither does mine. So over lunch, we developed this experiment that was supposed to be just between the U.S. and Croatia. And having sampled thousands of cankers at this site in Wisconsin, my hypothesis was that these thick areas of callus would be mostly comprised of the white form of the fungus. And if you looked at the area right on the interior, 9 and 10, 11 and 12, it would be things that weren't the chestnut blade fungus at all. That was my hypothesis going into this experiment. Go ahead. So she decides she's going to get folks from not only Croatia, but Switzerland and Macedonia to participate in this study that we developed over lunch. Go ahead. Here's my hypothesis, was that, and I don't want you to look at anything other than colors. Here's my callus. Here's the non-callus on the callus face. Is there more blue here than there is here? That's all I want you to look at. So this is Wisconsin. This is Switzerland. There's not a whole lot of white anywhere in terms of the blue for the white form of the fungus. But remember I said that most of Europe has been controlled by the hypervirulent form of the fungus? It doesn't take a whole lot because there aren't many compatibility types in Europe. You've got a whole stand of trees, you may have 1 or 2 compatibility types, that's it. Here we've got 30, 35. So it doesn't take much of the white form of the fungus in Switzerland to control the fungus. This is Croatia. A lot of white or blue. They got a good bit of hypervirulence going on there. And look at Macedonia. So the further east you go in Europe, the less hypervirulence you have. So Macedonia is much further east than Croatia. Go to the next slide. This is just a little bit of data. I just want to point out that here's my hypothesis of West Salem, 60 versus 39. So my hypothesis for West Salem was correct. It was supported. You don't get much in Switzerland at all in terms of the white. You get a little bit of significance here in Croatia and not much in Macedonia. Okay, so let's let me read that. I don't have time for that. I'm only on the second thing. I've got 2 more to go, so let's roll. So, Chestnut, are you going to talk about this breeding?
I would love to, or I could just say that my presentation isn't going to be that long, so you can go longer if you want, or I can talk about the breeding program if you want, but you can just keep going.
Okay, all right, keep going.
And
So,
talk so fast.
the Chestnut Blight, the American Chestnut Foundation was devised in the early 1980s based on this guy, Charles Burnham. Is he a chestnut blight breeder? No! He's a corn breeder. He got his PhD, his first job was in Morgantown at WVU in the early 1930s. What happened in the early 1930s in northern West Virginia? Chestnut blight's coming through. So he sees chestnut. He works on corn. He goes to Minnesota and spends the rest of his career in Minnesota. He's always thinking about genetics. Not necessarily in corn, but in other crops. And so he used a breeding technique in corn called back cross breeding. We're going to talk about that in a minute. So, keep going. So we're talking about crossing, American, Chinese. Keep going. I don't want you to go a lot of this, but the idea is to breed resistant Chinese and susceptible American, each time going back to American at each step. Each time, we're looking for form, a nice straight tree, and resistance to blight. What is that? That's that callus tissue that's walling that fungus off, okay? So what you do is you take a cross. What do you get? You get chestnuts that are germinating. You put those suckers in the ground, let them grow up to be 4 or 5 years of age. The ones that are real bushy, cut them down. The ones that have nice straight form, you inoculate with a fungus. If the tree dies, it didn't inherit any genes for resistance. If it calluses that thing off, that's what you want to carry on. So you do that each and every step. You're looking for form, you're looking for resistance. So now we're down here to trees that are 15/16 American and 1/16 Chinese. Go ahead. A lot of pollination. So where are American chestnuts? Are they across the street on a paved highway? No, they're out in the forest. So you've got to get cherry picker trucks up in there, and you got to get high up. And so what you do is you take those female flowers and you emasculate. You take all the male catkins off, so you don't get any pollination from other than what you put the pollen on, and you bag those to keep that. So when the fall comes and you take those nuts out of that, you know who's the mother and who's the daddy, okay?
So you're pulling the male flowers out and putting the male flowers of what you want in?
Onto the females, yep, on
Like
Americans.
a paintbrush or something?
There's different ways of doing it, but I mean, we can pour pollen into a cap and then dip the female flowers into it. Some people use a glass.
It's all hand-made.
It's all done manually. Well, not always. Sometimes we use a pollen sprayer. We get those wild trees and put it up at the top of the tree, blow clouds of
And
pollen.
then you bag it.
All this work, where the trees are grown, is in Meadowview, Virginia. So if you look at Virginia, that little tip that comes down right above Bristol, Tennessee, that little skinny part of Virginia, that's where these are growing. So on I-81, and exit 24 is Meadowview, Virginia. We've got 120,000 trees that have been planted over the course of time. And we're gonna cull this down to about 500 of the best, right? So we're looking for 120,000 trees down to 500. Okay. How do we look at that resistance again? Okay, we take a little punch up with the orange form of the fungus in. Okay. And then what we're looking for at Meadowview are reactions to resistance. This has got a good form. Look at this canker. It's nice and calloused over, you don't see much. This is an intermediate, you're not going to keep this. And you sure as heck aren't going to keep that. So you can look at the trees that have been produced over this breeding program and pick out the ones that look like this. You don't want these or these. And these are the ones you're going to continue on in the breeding process. At WVU in Morgantown, we've got 900 trees planted, pure American, pure Chinese, pure European, and a lot of these back crosses. So these little twigs I passed around came from here yesterday. Okay. So what we did at Morgantown was we had 6 blocks of 150 trees each. That adds up to 900. 3 of them we treated with the punch and the hammer and the white goop. The other 3 we didn't do anything with at all. So we're looking for differences in whether the white form of the fungus, we call this a marriage experiment. We're marrying hypervirulence and increased resistance from the tree. So does that white form of the fungus help when a tree has increased resistance or not? Okay. I used a subjective 0 to 4 scale. Does the canker look like this? Does it have callus tissue, but yet it has those orange bumps? This has no orange bumps at all, just callus tissue. This has a good bit of callus, as you can see, but there's a lot of orange bumps. That's a number 2. A number 3 is just a little bit of callus and all orange, and a 4 is lousy. It's not, there's no callus at all, it's just, so the lower the number, the better. The higher the number, the worse. Okay. Just a little bit of data. So here's our Americans, our back crosses, our Chinese, and our Europeans. Take-home message is percent living, 69 to 47. So the fungus helps Americans, 78 to 67. It helps this back cross, 69, 49. It helps this back cross. Doesn't do anything for Chinese because they're already resistant. Europeans, 51 to 15. So with the exception of the Chinese, the white form of the fungus enhances the percentage of trees that are living. That's not the whole story. The other story is a tree can be living, but is it vigorous and growing great, or does it look like crap? You know, it's almost half dead. So I mean, you have to look at it in a realistic way. And so here's my rating. Remember, the lower the number, the better. The higher the number, the worse. So are we any better than Americans? No. Are we any better with this back cross? No. Are we any better with this back cross? No. Are we any better with the Europeans? Yes. So, even with Americans, so we're talking about Americans and Europeans benefiting from hypervirulent treatment. The back crosses in the Chinese, not necessarily. So the bottom line is, all the species and hybrids, with the exception of the Chinese, benefited from treatment with that white form of the fungus. After 3 years of treatment, tree health, the hybrids and the Chinese were uninfected. There wasn't any difference in how a tree looked in terms of its looking good or poor. So the bottom line is that the TACF's breeding program, those trees seem to have acquired sufficient resistance to make them look better without the white form of the fungus. So the bottom line is the breeding program seems to be working. That's the bottom line. Okay, let's go. Pennsylvania is one of the chapters where New Jersey have lots and lots and lots and lots of plantings of all kinds of trees, chestnut related. Go to the next slide. West Virginia, we have a number. This number is 42. It's going to go up to at least 45. Where we've got trees from up at West Liberty University, clear down here into Wayne County. How many of you knew there's a Russian Orthodox monastery in the state of West Virginia? Oh, you knew that. You knew that too? Okay, in Wayne County, so we've got trees planted all across the state. Okay? Last thing I want to talk about. So we talked about hypervirulence, we talked about breeding, now we're going to talk about transgenics. So at the State University of New York, SUNY, State University of New York. They're doing genetic engineering. You all, most of you all eat cereal, right, for breakfast in the morning. Many of you eat Wheaties or something. You eat white bread. It's made out of wheat. Wheat contains an enzyme. Remember an hour ago we talked about oxalic acid? Wheat contains an enzyme, oxalate oxidase, that breaks down oxalic acid. So they're taking that gene from wheat and putting it into the American chestnut tree. What's oxalic acid do? Or oxalate oxidase do? It takes oxalic acid, breaks it down into hydrogen peroxide, you know that stuff you pour on your cuts, and CO2. That's it. So it's not a pesticide. It's a plant protectant. So it's not producing anything that kills the fungus. It's simply reacting with the enzyme that goes to form peroxide and CO2. It reduces the chance of the fungus to develop resistance to oxalate oxidase, and it changes the orange form of the fungus from a pathogen to a saprophyte. So, they have developed a small stem assay. So a lot of times, remember I talked about the fact that you got to take these little germinating nuts, grow them up into a tree that's 7, 8, 10 years old, and then inoculate them. They developed a small stem assay. So these things are, you can tell, they're about the size of his thumb. So these are 2 years old. They developed a small stem assay where you take a scalpel, make a little incision, put that form of the fungus in here. Go to the next slide. These are Americans. These are Chinese. And these are some of the transgenic trees developed at State University of New York. Remember that wilting we talked about really early on? The flagging? These little leaves have all wilted. We even have some wilting here with the Chinese. These have the oxalate oxidase gene. And they're pure American, and there's no wilting at all. Take a close-up of one of those stems. This would be an American stem. After you put that little slit of the fungus in there, let it grow. Here's that tree that has genetically engineered gene from wheat in it. Look how small it came. It's already been into callus over, and it's only just not very old. Now, let's say you produce a lot of trees. How do you know whether your tree has the oxalate oxidase gene or not? They've developed an assay where you take a punch. You know how you use the punch for paper? Take a punch, add a chemical to it. If it turns black, it's got the oxalate oxidase gene. If it doesn't, it didn't inherit that. So when you take pollen from a transgenic tree and you pollinate a flower on a pure American, and you get a bunch of nuts to grow up, half of those are going to be containing the gene, and half will not. And this is a quick and easy assay to determine early on whether your tree does or does not have that gene. Okay. Final note. This is from an article in New Forests Journal in 2017. It said the chestnut restoration effort is the most ambitious, protracted, and technically difficult rescue of a species that has ever been undertaken. Think about that for a minute. The most technically difficult restoration of a tree species ever. So the American Chestnut Foundation, of which I said there are 16 state chapters, are doing what we can to restore, not in my lifetime, or my children's lifetime, or my grandchildren's lifetime, this tree. But the hope is that we can, at some point in time, develop and put these back into the eastern United States. That breeding program, they hope, would be completed in 2020. I don't know if that's still right or not. But
Yes.
this started here. The New York transgenic trees still much undergo regulatory review. You got to go to the EPA. You got to go to USDA. FDA is what's the word I'm looking for? Not mandatory, but optional. Okay. Research and ideas probably 5-6 inches thick of documents to be submitted to the EPA and USDA to release this transgenic tree. Right now, you're in a 10-acre block, and when these trees flower, you've got to emasculate them all. Take all those flowers off, because you don't want anything until it's approved, going from a bee from a transgenic tree up the hollow there in New York State to pollinate another chestnut tree.
What's the fear of what could happen?
Okay, so Tom can talk about that better than I can. But the idea is that as a university in Syracuse, you're developing this tree. You have to answer questions of the EPA and the USDA that they haven't asked, but you have to develop. So, for an instance, how does it, since this is many times a riparian or a stream tree, how do the